Hello, everyone, and welcome to today's CryoEM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for CryoEM Access and Training, NCCAT. I'm joined today by my colleague from NCCAT, Ed Eng, along with Michael Schmidt from the Stanford SLAC CryoEM Center, S2C2, and Vamsi Rayaproilu and Nancy Meyer from the Pacific Northwest CryoEM Center, PNCC. PNCC invited today's speaker, Dr. Camden Driggers, who's joining us from the Oregon Health and Science University in Portland. And Nancy will introduce him properly after Michael, Ed, and Vamsi give us a short update on the resources that we make available at no cost to the research community. We'll all of us here on the panel today will be here the whole talk. So if you do have questions about how you become a center user, feel free to send those questions to the panelists, either through the chat or the Q&A, and let us know if you aren't a current center user and want to be, let us help you figure out how to do that. This is a regular event. We host the last Thursday of each month. Our generous users share the methods and highlight tips and tricks that have enabled them to collect and interpret cryo-EM data with the help of National Center resources. So you can save the date already for our September talk. Bradley Gilliams from Colorado State University will be giving a talk on using cryo-EM to characterize a clonable selenium nanoparticle. And the registration link for that talk will be available soon on the events page at cryoemcenters.org, where you can also find recordings of all of our previous talks. Back to today, though. If you have questions for the speaker, please send them at any time using the Q&A box. We'll look for them there, not in the chat. And Nancy will moderate those questions at the end of the talk. So let's go ahead and get started. So first, some updates from the centers. And Mike, why don't you let us know what's new at S2C2?
Hi, I'm Mike Schmidt, and I'm one of the co-directors of Stanford SLAC CryoEM Center. We have four Titan Krios microscopes, and we offer, of course, high-resolution data collection, training, and screening facilities for our users. We're very excited for this next month to have a very large in-person workshop hosted by SCSC, featuring Ali Punjani and his team with CryoSpark developers, and Ellen Zhang for using CryoDragon. We have 75 people that are coming, and it's going to be fun. There's going to be a room for 100 or so more people joining on Zoom, but it's going to be a great time, September 16th through 18th. We're looking forward to it.
All right, thank you, Mike. Ed, why don't you let us know what's new at NCCAT?
Well, NCCAT offers high-resolution data collection, screening, and cross-training services. I think everyone had a good summer season, and now it's workshop season because we will be holding an integrative structural biology symposium October 9th through 11th. That is almost full, but we will have a workshop Wednesday with 8 workshops. We'll highlight the TFS Glacios G2, which we just commissioned over the summer with TomoLive, Smart EPU, and Multigrid. Gatan will demo their Alpine Vista. SPT LabTech will have a workshop on the Chameleon. And just quickly on the others, Mitogen and Nanosoft will show off their plunge freezing and grid clipping hardware. Nanotemper will show their protein stability characterization. Prometheus Syncell Microscope will be there for the precision photo labeling. Avioli will demo their Primo micropowdering unit. And finally, Schrodinger will give you a PyMol3 visualization tool workshop. So if you want to find out more, go to nccat.nyspc.org.
Thank you, Ed. And Vamsi, why don't you let us know what's happening at PNCC?
Hello, everyone. My name is Vamsi, and I'm a microscopist at PNCC. PNCC accepts 2 different types of proposals, screening access and a general access. Screening access allows you to come on site for up to 5 days of sample preparation and screening with the staff. This proposal is aimed at low resource institutions, especially ITS state institutions. General access proposal awards up to 480 hours of microscope time a year for 2 years. Hours. That's a lot of time. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops. We actually did just one today last week. It was called the CryoEM school that lasted for 2 weeks. We went through the whole process of screening, grid preparation, screening, and data collection. Approved proposals are also assigned a scientific point of contact, a SPOC, if you may, who will assist with questions about sample optimization, data collection strategies, and so on. We have an Arctica and a Glacios, newly installed Glacios 2 and 4 Cryoses. 3 of our Cryoses have K3 cameras with biocontinuum HD filters. One of our Cryos has a Falcon 4i with a Selectress energy filter. The Glacios 2 also has the Selectress energy filter. And for sample preparation, we have a Vetrobot, like a GP2, Vetrojet, manual plungers. So if you're interested in applying, our deadline is the 1st of every month. We definitely look forward to hearing from you.
All right. Thank you, Vamsi. I'll stop sharing and let Nancy introduce the speaker.
Unmute myself. We're very happy to have Camden Driggers here with us today. A little bit of background. So Camden received his bachelor's degree in chemistry, cell bio, and neuroscience at Montana State. He then moved to Oregon to pursue his graduate work with Andy Carplus, at Oregon State University, where he led several successful X-ray crystallographic studies. His experience then includes solving high-resolution crystal structures, focusing on small molecule active sites and kinetic studies. In 2020, Camden came to OHSU and joined the lab of Xiaoling Xing, who's a world leader in potassium ATP channel structure in the School of Medicine here. He's learned CryoEM and has been contributing to defining ligand-mediated mechanisms that regulate the ATP-sensitive potassium channels, looking for clinically significant drug interactions and natural ligands. So he is going to share some of that work with us today. And with that, I will hand it over to Camden.
Nice. Thank you, Nancy, for the introduction and also for inviting me to speak here today. It's a great opportunity because the Xing Lab has been working with the PNCC since it started as a national center, which is pretty cool, starting with former lab members Greg Martin and Minmu Song, and working with Nancy mostly as our SPOC with help from Craig and Irina and many other helpful staff scientists over the time since 2017. And so today I'll tell you about just one recent structural study from of the KATP channel complex with some background as well. And to start off, let me just introduce the KATP channel. The KATP channel is a complex of 4 inward rectifying potassium channel subunits that form the core that actually is the potassium channel right there in the center, and 4 sulfonylurea receptors, which are named after the drug, with the receptor being identified later after. These sulfonylurea receptors are a classic ABC type transporter family with the TMD1, TMD2, and then the 2 nucleotide binding domains, 1 and nucleotide binding domain 2, which are also shown in the structure down here, nucleotide binding domain 1 and nucleotide binding domain 2. And these bind to nucleotides and come together and dimerize, or being not bound to nucleotides and separate. And it also interacts with, here's a sulfonylurea globanclamide bound to the channel. These ABC type transporters, these sulfonylureas, are unique in that they have no transport function. A typical transporter would be transporting a molecule between the 2 transmembrane domains, but SUR does not do that. Instead, it also has this unique TMD0 domain that interacts with the inner rectifying potassium channel, and it serves to regulate KIR channel. Today, I'll be talking specifically about the pancreatic KATP channel, which is a fusion of KIR 6.2 and SUR1 channels. I wanted to point out, just while we're on this slide, a couple of features. The KIR 6.2 has a cytoplasmic domain that's actually made of contributions from both the N and C terminal. And it binds to nucleotides, ATP, in an inhibitory way. And the C terminal has a ER retention motif, the RKR. SUR1 has this ICL1 loop that's important for interacting with the KIR channel that I'll talk about later. And then I mentioned already the nucleotide binding domains as well. And so in pancreatic beta cells, KATP channels couple insulin release to metabolism. So in a resting state with low glucose, ATP levels are relatively low, ADP levels are relatively high. And in this state, the potassium channel, the KATP channel is open, which means the beta cells in the pancreas are hyperpolarized. The voltage-gated calcium channels are closed and no insulin is released. After a meal or with high blood glucose conditions, the glucose is metabolized to ATP, which leads to relatively high ATP levels, relatively low ADP levels. And this closes the potassium channel, depolarizing the beta cell, leading to the opening of voltage-gated calcium channels, influx of calcium, and release of insulin. Congenital hyperinsulinism is caused by loss of function mutations in the KATP channel, which drive it towards this open state. And gain of function mutations cause neonatal diabetes and Den syndrome, which is a developmental delay, epilepsy, and neonatal diabetes syndrome. KATP channel gating is controlled by ATP binding to the inhibitory site on the C terminal on the cytoplasmic domain of KIR 6.2. And in this closed state here, you can see the NBD nucleotide binding domains of SUR1 are separated, and ATP is bound to KIR 6.2. Magnesium nucleotides can bind to the NBD1 and NBD2, and also PIP2 is important, and it can bind to the channel and push it towards the open conformation. A lot of our historical studies in the lab have been through patch clamp recordings, electrophysiological recordings, where you can take a portion of the cell and take a patch from it and do these kinds of recordings. And so these are cells that are under physiological conditions. And so you'll extract the patch with whatever kind of native lipids that it currently has. And so for a wild type channel, you can see if you extract the patch initially, your currents, this is totally closed here, and then this is like some open. And when you add ATP at a couple of different concentrations, you can see initially both concentrations inhibit the channel, both 1 millimolar and 0.1 millimolar. But with repeated additions of PIP2, it opens the channel further. And as you add more and more repeated additions of PIP2, you can see that initially the 0.1 millimolar effect nearly goes away, and even the 1 millimolar effect gets diminished quite a lot over time as you kind of load the channel more and more with PIP2. And I'll show some electrophysiology recordings like this, similar to this throughout the talk. Also, this electrophysiology recording above here shows the channel initially open, and when you add ATP, closes the channel. A little bit less ATP as the channel slightly open, but when you include magnesium ADP, it has this NBD dimerization event driving opening of the channel, and then again, the same on the other side. And so for wild type KATP channels, for this recording, there's kind of closed on the bottom and open on the top. And we see an open probability of like about 0.6 for a wild type channel. And an IC50 for ATP inhibition is about 10 micromolar. Interestingly, if you delete the N-terminus of KIR 6.2, about 30 residues of the N-terminus, the open probability increases and the channel is much less sensitive to ATP inhibition. If you delete the C-terminus of KIR 6.2, which is deleting that ER retention RKR motif, which allows the KIR 6.2 channel to express to the surface by itself, which typically it wouldn't do. Its open probability is quite small, and the ATP inhibition is quite a lot less sensitive than the wild type channel. If you take those delta C channels and express them with SUR1 TMD0 domain only, the PO returns to close to wild type, but they remain much less sensitive to ATP inhibition. And really what we're trying to answer with this talk is like, what are the underlying structural mechanisms for these phenomena? And getting ahead a little bit, we reviewed a number of different KATP channel structures that have been solved by our group and 2 others over the years. And these structures include ones that are bound to sulfonylureas, inhibitory ATPs to the C-terminal domain of KIR 6.2, and then also ones bound to magnesium nucleotides and dimerized. And our questions are, how does SUR1 increase the ATP sensitivity of KIR 6.2? How does the N-terminus modulate channel open probability and ATP sensitivity? And how does SUR1 TMD0 increase the open probability of KIR 6.2? And so in 2019, Greg Martin and Min Woo published a structure showing the CryoEM structure from PNCC collected data, or at least the microscopes there. It may have been before the center was officially PNCC, showing the interaction of the KIR 6.2 N-terminus with SUR1. And so you can see here the KIR 6.2 tetramer and the N-terminus extending in between TMD1 and TMD2 of SUR1 and interacting with it. And if you look down below and you see the wild type channel recordings compared to the KIR 6.2 with the N-terminus tail deleted, you can see that there's a much higher open probability of the channel when that N-terminus is removed. So it has an inhibitory effect. And actually, the sulfonylurea drugs bind just adjacent to the N-terminus of KIR 6.2 and help stabilize it inside the cavity between TMD1 and TMD2. And to do some biochemistry on this, we took a residue on SUR1 and mutated it to a cysteine, and took a corresponding residue on the KNT of KIR 6.2 and mutated it to a cysteine, and allowed a system that was controllable by oxidation and reduction to either lock this N-terminal domain in the cavity between TMD1 and TMD2, or to allow for its release. And so we can see initial currents. When you add the inhibitory 1 millimolar ATP, the channels close. And then when you when you take it back out of that solution and then expose it to iodine, it slowly oxidizes, causing the channels to close close more as that cysteine disulfide is formed on the channels. And then if you move it to a solution of dithiothiazole, DTT, you can see that it's breaking that disulfide bond, allowing the channel to open more and more, moving it back into ATP, and then again, oxidizing with iodine to close the channel and reducing with DTT to open the channel. This leaves us with an interesting mutation that these closed channel structures didn't yet uncover. And this is a neonatal diabetes DEND causing mutation that's found in human patients called Q52R of KARE6.2. And so for a wild type channel recording, moving into different concentrations of ATP, the channel is fully inhibited at 0.1 millimolar ATP and close to half inhibited at 0.01 millimolar ATP. But for Q52R, it's almost tenfold less sensitive to ATP, with 0.1 millimolar being about half inhibition and 0.01 millimolar barely inhibiting the channel at all. And you can see the IC50 difference between Q52R and wild type channel is something like on the order of 6 micromolar to 125 micromolar. If we look on the right-hand side of this slide, you can see the wild type channel with open and close compared to Q52R. And the Q52R is much more open. But this, interestingly, the effect of this Q52R mutation is SUR1 dependent. If you take those KARE6.2 channels and delete the C terminus so that they can be expressed to the surface, and you have them wild type otherwise, you can see that the trace is quite similar to when you have that Q52R mutation present. So it's an SUR1 dependent mutation that's present. And a mutagenesis screen tried to search for that interacting partner in SUR1. And the long story short is that no such partner was found, but interestingly, there was a partner found that locked the channel in a closed conformation. So this Q52E with this K205 can really like lock the channel into this this closed conformation. And so it's still unknown how Q52R opens the KATP channel and drives neonatal diabetes. And so that's going to be the subject of my talk today is our structural studies to solve the open channel structure of the Q52R full channel. And so I wanted to take a little bit of time to walk through the workflow because I know that this audience in particular is interested in this. And so we do our expression in mammalian adherent cells, either INS1 cells, which is a pancreatic beta cell line from rat, or in COSM6 cells, which are mammalian adherent cell line that does not express endogenous KATP channel. And we use these COSM6 cells when we're doing any kind of mutation study so that we don't have mixing with wild type channel. These are transiently transfected with adenovirus. And it's a separate adenovirus for each of the genes coding for KARE6.2 or SUR1, and then also transcycline transactivator gene. We require about 10 15 centimeter plates for one cryo-EM prep that gives us anywhere between 6 to 10 grids, something like that. We take the cells, prepare a membrane fraction and solubilize it in detergent, digitonin or GDN, and do a flag affinity purification. And the flag tag is only on the SUR1. the extracellular side of SUR1. And this allows to purify KATP channel particles in detergent micelles. And you can see denaturing SDS page gel here with SUR1. It exists in two forms, a core glycosylated and a complex glycosylated. The core glycosylated is before it has exited the ER and traveled through the Golgi. And the complex glycosylated is what we think of as the surface express channel that has already passed through the Golgi and traveled onto the plasma membrane. And then a native gel as well, showing the full channel complex and some dissociated channel. A negative stain of a typical concentration of the KATP channel particles is shown here. And then a CryoEM micrograph over just ice shown here. We've tried a number of different other surfaces, including lacy carbon. You can see the different KATP channel particles are circled there. And then also Minwoo Sung in our lab worked on developing a technique for making in-house graphene oxide coated grids that give a great advantage, but also do contribute to heterogeneity of the grid. So from the same grid, we can see this beautiful micrograph with many particles, but then also there are some areas that have much too thick geo. And so you have to be kind of more selective in your data collection strategy when using geo, but it does provide advantages when it works well. And this is sorry about that. This is one of two different data processing workflows for this that yielded equivalent maps. And I'll show the other one later. This one is using a kind of extensive 2D classification to get a good number of particles. So only like nice, good looking 2D classes were selected, about 70,000 particles. And then subsequent rounds of cryospark ab initio reconstruction of different numbers of classes to whittle down to only those particles that were contributing to the higher resolution reconstructions. And that left a relatively small number of particles, about 15,000 particles, but that refined to about 2.9 angstroms resolution with a mask. And a local resolution, you can see it's much better defined around the core and TMD0 of SUR1, with the end of SUR2, especially TMD, especially MBD2 is much more highly mobile. And these maps revealed an open potassium pore and a separate nucleotide binding domains. And so typically we think of the nucleotide binding domains as being dimerized as to help drive in the opening of the channel. And so I think that's one of the reasons there's such high mobility and high flexibility in MBD2. During this protein purification, I included brain PIP2, which is a long chain PIP2 with the membrane fraction. And we were able to see, surprisingly, we're able to see lipid density that we assigned as two adjacent PIP2 molecules between KIR 6.2 and SUR1. And all inward rectifying potassium channels have a conserved PIP2 binding site that's known to regulate function. But this second site shown in purple is novel, or I mean unique, at least among the inward rectifying potassium channels, and is formed with contributions from SUR1 as well. There's other interleaflet lipids visible besides the two modeled KIR 6.2. So I'm sorry, the two PIP2s. And so we have, you can see the purple map is for that PIP2, and here's it just shown at a couple of different orientations, rotated by 90 degrees. And then the cyan is the canonical first site, shown in a couple different orientations as well. And then From the same orientation, we also are able to model a phosphoserine, which is just endogenously purified from those COSM6 cells. And then also we also able to purify the channel with these endogenous lipids on the other side. So this is rotated 180 degrees. And you can see there's phosphoserine, phosphatidylethylamine kind of stacking. Here's them looking at them as two separate, but they're adjacent and seem to be interacting with each other. I wanted to show some just representative map to model fits, because it's hard to show this when you're looking at the broader picture. We have the KARE 6.2 selectivity filter, which is actually what allows these potassium channels to distinguish between other ions, especially sodium and potassium. And it's at each one of these sites, two of which are occupied by a potassium in our model, but there's a site here as well. And I'm just showing two of the four subunits. The ones facing us and away from us are not shown. But these oxygens, either from the threonine here or these carbonyl oxygens, are mimicking the hydration site of the potassium. And so a hydrated potassium will be coming up here, lose its waters, and then kind of hop along the hydration sites, pick up more waters here on the other side as it exits the pancreatic beta cell. Here is part of SGR1 ICL1 forming a beta sheet with the N terminus of KARE 6.2 residues about 44 to 48, and then the start of the continuous beta sheet of the cytoplasmic domain of KARE 6.2 shown here. Here's another region of the KARE 6.2 CTD. And the M2 gating helix of KARE 6.2, when of course all the transmembrane helices have very nice density, and the side chains are able to be well defined within those especially. And so the overall structure of these open and closed KATP channels, we have the pore path shown here, and you can see relative to the closed structure down below, there's a widening at the helical bundle crossing gate, and the two PIP2 molecules bound. And the CTD of KARE 6.2 is rotated up and further towards the membrane, which is characteristic of inward rectifying potassium channel gating. And in the closed structure below, both of these being MBD separated, but in the closed structure below, you can see ATP bound to the C-terminal domain of KARE 6.2 and glibenclamide bound in between TMD1 and TMD2 of SGR1. Here's a close-up view showing the opening of the inner helix gate, or the helical bundle crossing, we also call it, with this phenylalanine 168 residue really being key for swinging away from the pore and allowing it to open up. And it opens to something like 3.1 angstroms, which seems to be enough to allow the hydrated potassium ion to pass through. In the closed channel structure, the M2 helices are rotated slightly and more parallel to each other, and the F168 is rotated towards the pore and constricting it. And we can look at that with a plot, with the gray being the closed channel structure and just the potassium pore radius. And you can see that it's quite closed in the closed structure with those phenylalanines blocking it. And then open here in the inner helix gate. And there's kind of three constriction points shown here. The selectivity filter, which actually contains the oxygens, so it allows the potassium to go through it, but it's a non-hydrated potassium ion. And then the hydrated potassium ion has to pass through the inner helix gate as well as the G-loop gate, which in both open and closed are in a similar semi-open state. Here's a morph between the ATP bound closed structure and the PIP2 bound Q52R open structure. And this shows nicely the kind of the closing of the inner helix gate here. And then also I wanted to point out, which I'll talk more later about, but the tryptophan 51 undergoes this large conformational change. And this is at the TMD0 interface with KIR 6.2. And this residue that has no head group here is the Q52R. So you can see it's flipping around to kind of interact with the tryptophan 51 in the open structure, but is very far away from it in the closed channel structure. A close-up view of that interaction, Q52R interacting with tryptophan 51 with some CryoEM maps shown. And also a rearrangement of this SUR1 ICL1, which is a helix in the closed structure, becomes part of the beta sheet of this continuous beta sheet of the CTD of KIR 6.2. When we do some pairwise mutations, we can see that the Q52R plus W51C kind of abolishes the opening channel effect that Q52R brings, with and restores it to something more like wild type. And we show that both with electrophysiology and also with rubidium efflux experiments. This is a rather busy figure, but I wanted to show it because you can see the phenylalanine 168 gating residues here, and I'm not showing the fourth down off the screen here, in density, as well as tryptophan 51 and Q52R in the background here, with the two PIP2 molecules. And this panel is looking from the outside of the cell in, sort of like almost down through the pore, the potassium pore, and then rotating at 90 degrees. So now we're looking from the plasma membrane into the side of the channel, with the second PIP2 site shown here, and the first canonical PIP2 site shown here. And this lysine 134 contributing to the first PIP2, or I mean the second PIP2 binding, but not to the first. And so this SUR1 K134 contributing to the second binding, we tested using mutagenesis followed by electrophysiology. And so when you first have your wild type channels extracted from the patch, the current is relatively high because presumably they're bringing some endogenous PIP2 with them. And then adding PIP2 allows them to open more, but that effect is much more dramatic when you're using the K134A mutation, where the currents start quite small and you have to have repeated additions of PIP2 to kind of charge the channel and be able to open it of high concentrations of PIP2. And so the current increase in PIP2 is much higher for K134A, since it's starting off kind of more apo and you have to charge it with a lot of PIP2. Interestingly, there are a number of different like hot spots for human neonatal diabetes and DIN syndrome and congenital hyperinsulinism nearby both of the PIP2 binding sites. And so the circles shown in green are kind of hotspots for neonatal diabetes. And so that means their gain of function mutations, leading to the channel being more open. And then the yellow circles are hotspots for congenital hyperinsulinism, meaning that they are making the channel more closed. And then there's one that's a mix that is depending on what it's mutated, what residue it's mutated to. The opening of the KATP channel, interestingly, decreases the overall curvature of the membrane. You can see in the closed channel structure, it has this kind of more domed shape along the membrane. And when the channel opens, it's much more straight. And driving that is a rearrangement at the interaction between TMD0 and KR6.2. And so the closed structure is the gray, is shown in gray, and the colored structure is the open channel structure. And you can see the helix interacting with KR6.2 is shifting over, especially at the cytoplasmic side of the channel, and relatively fixed in the surface. So it's kind of like driving this like straightening of the overall channel. It's always nice to, when you have nice maps, to show density. So here's the density of that interacting helices in the open channel structure. And then again, the closed channel is shown in gray, and then the opened is colored. And you can see the dramatic motion of tryptophan 51 that we've observed in previous slides, as it kind of swings out and driving this like overall straightening of the membrane. There's this rearrangement of the SUR1 ICL1 that I've mentioned previously, from being this helical structure that's not interacting substantially with the continuous beta sheet of this of KR6.2 CTD, but in the open channel structure, it becomes part of a continuous beta sheet through the structural arrangement, and you can see the Q52R interacting with tryptophan 51 here. Looking from the outside of the cell in, and here's the potassium pore, and then the potassium pore closed with the phenylalanine 168 blocking it, and then the phenylalanine 168 nicely open. And this is to illustrate also that rotation of the C-terminal domain that's happening between the open and closed structures, which is indicated by this kind of red arrow, and this is just a point marker, so you can kind of keep track of the rotation. And so we can, to put these motions all together, it's like the overall straightening of the membrane, the rotation of the CTD between open and closed, and also this rearrangement at the interface between TMD0 and KR6.2 involving tryptophan 51 and Q52R. I wanted to talk about an alternative 3D classification strategy that allowed us to parse out an APO closed conformation from within this same data set. And it's much lower resolution, as you can see. So the yellow classes are those open channel classes, and the blue is the closed channel class. from within the same data set. And this is using the same particle set from 2D classification that I mentioned in the previous workflow. And so for the open, you can see that the resolution is quite a lot lower than for the closed. And so they were just being tossed out by the previous strategy. But actually, it's an interesting effect because the open structure has the KATP absent from between the TMD1 and TMD2, but in the closed structure, that KATP is actually bound, and we can still see it. And so that's an interesting effect because it means that MBD dimerization is not the only thing driving the dislocation of the N-terminus from between TMD1 and TMD2, but actually that channel opening requires the N-terminus to be disassociated from TMD1 and TMD2. In the open channel structure, I've also indicated the KIR 6.2 CTD is up, rotated towards the membrane, and then in the closed is down and disassociated away from it. And then relative motions for the SUR1, with the SUR1 being relative, the MBD being relatively closer, and the relatively further away in the open. A close-up view of that, you can see from the side view, looking from the side from the plasma membrane and looking from the cytoplasm outside of the cell, you can see the rotation of the CTD as well as the shift in the MBD1 and MBD2. And to kind of put that all together in this video, let's see if that plays. There we go. You can kind of see the transition in between the open and closed channel with that rotation of the CTD and the large motion of SUR1. That's great. I'll play one more time to illustrate also the tryptophan 51 flipping around. And all of this is driving the rotation of the gating residues in KIR 6.2. A close-up look of the lack of the N-terminus of KIR 6.2 with the N-terminus of KIR 6.2, which we call the KNTP bound between the PIP2 bound open and what I'm calling the APO closed. And in fact, the open structure, which is the two panels shown in A, and these are just shown at two rotation. It's just a surface representation of the atoms shown at two different rotations so that you can see the first canonical site, PIP2, and the second site fitting nicely in the open structure. But in the closed CTD up RPG bound structure, you can see the first site could be occupied, but the second site is blocked. And then this C panels are the APO CTD down closed structure that was parsed out from this data set. And you can see again, the open, the first PIP2 site is bound or could be bound. And the second one is blocked by the protein. And so with that, I'll finish the talk by summarizing. And in the closed channel structure, we just have the KNTP inserted between TMD0 and TMD1 and TMD2. And with KNTP inhibitors, like sulfonylurea drugs, stabilizing that interaction, and ATP bound to the C-terminal domain at the inhibitory site of KIR 6.2. And then we can have open form in two different ways, and we think that including PIP2, having the Q52R mutation allows us to have this open structure of the carrier 6.2 that matches or mimics the open structure that's also induced by MBD dimerization as well as PIP2 addition. And in both of those cases, the N-terminal domain of carrier 6.2 has to be disassociated from the cleft between TMD1 and TMD2. And this can be driven also by KATP channel opener drugs, such as the clinically used diazoxide, and showing MBD dimerization between the magnesium nucleotides and the two PIP2 molecules shown here. And with that, I'd like to acknowledge everyone, especially acknowledge my mentor, Shouling Xing, and Minwoo Song, and Greg Martin, who did a lot of the, or did the early structural studies in our lab, and then of course everyone else in the lab contributed to all of the functional studies that I talked about today. And then our collaborators lab, Dan Zuckerman for doing many of the MD simulations that have been important to insights that I didn't get in, but some of the insights I made were based on that. And then also Colin Nichols' lab, especially Sanju, for helping with some structural work and for sharing constructs with us and things like that. And then, of course, I would like to thank the Pacific Northwest CryoEM Center, Nancy, our Spock, and Craig for giving helpful insights. And then the OHSU multi-scale microscopy core, especially Stephen Aminu for teaching me how to use my first CryoEM microscope almost 5 years ago now, I guess. And then, of course, our funding. And with that, I'm happy to take any questions. Thank you.
Great, thanks, Camden. We've got a number of questions about the science that you can field. So first off, we'd like to know, could you please comment on the brain PIP2 and the soluble DIC8 PIP2? Did the brain PIP2 work better than the DIC8 PIP2 in cryo-EM, even in the presence of detergent?
Yes, definitely. So that's a great question, actually. And I wanted to go back to my very early slide, kind of showing the electrophysiology charging of the PIP2, which uses long chain brain PIP2. And so the long chain brain PIP2 is much more soluble, the long chain brain PIP2 is much more likely to like stay in the membrane and stay bound to the channel than the soluble 8 carbon chain short chain PIP2. And so this is with the long brain PIP2, and you can see that with repeated additions is actually adding more and more brain PIP2 to the channel. That's actually like kind of charging it over time. And so other groups have tried to use short chain PIP2 to capture a structure of the PIP2 bound complex, but I think by using long chain PIP2 before detergent solubilization, we were actually able to like trap it in the detergent micelle and then be able to visualize it after collecting data on those particles. That's a great question.
Thanks. And another question, could you explain how the PIP2 reduces ATP sensitivity? Does PIP2 binding directly affect the ATP binding?
And it does. And if I can, let's see if I have the nice slide that shows. And so there are residues that actually contribute to both PIP2 binding and ATP binding. So you could, with ATP bound, it's in a conformation that won't accommodate especially that second site PIP2. And so in the ATP bound form, you have to have PIP2 bind in order to, you have to have like this kind of induced fit. So you have to have higher concentrations of PIP2 to cause the channel opening. And in the PIP. When PIP2 is bound, you have to have like a lot more ATP present in order to like pull the residues away from PIP2. So it's like it is a direct competition. And I think I have a nice slide. Some of the residues, like you can see this Q57 actually is one that will actually be interacting with ATP or up towards interacting with PIP2, and there's a number of residues like that too.
Thank you. And we have congratulations on the publication, and it really is a lovely structure. Two questions. One is dynamics of TMD 1-2 of SIR1. Did you get a chance to see the TMDs, NBDs getting close by 3 dBA?
Yeah, that's a good question. If I can look at the, that's a sharp question actually. So there is a lot more mobility in especially NBD2 and TMD2, and so it has like a moving towards dimerization kind of effect. Like it's not dimerizing, but the mobility is much higher than for our closed NBD separated structures. But I wasn't able to see like it with 3D variability analysis, wasn't able to see it like fully going to NBD dimerize, but it was it was towards it certainly. And yeah, the lower resolution of the NBD2 is kind of like in these maps kind of showing like how mobile that really is. And I think the reason for that is because this open channel structure is predispositioning it towards NBD dimerization, just like NBD dimerization is driving the opening of the KATP channel. Like having this open channel structure with PIP2 bound is predisposing the structure towards that NBD dimerized, and so it makes that NBD2 much higher, much more mobile. And I think I think a follow-up study would be doing the structure under NBD dimerization conditions would be really interesting. And then I think if we were able to, I think if I had more particles, also that we might be able to see that motion in 3D variability analysis better. That's a great question, though.
Thanks. And there was a second
Nancy,
part.
if I can interrupt you for a
Sure.
second, just because we are coming up on the hour. Camden, if you're willing to stay on another couple minutes to finish answering questions, is that okay for you?
Yeah, that's okay.
All right. And for those of you who have to run off, we're going to keep recording. So if you need the last few questions, come find the recording. And we hope you join us again next week. And for those of you able to stay on, Camden will let you finish answering some questions. Go ahead. Thank you.
I think we've just got two more for you, Camden. The CTD rotating up might have affected the KNTP. Do you have any guess or hypothesis on where the KNTP would be located?
You know, I think that's a great
more specific question. So you might want to like explain a little bit.
Yeah, that's a great question. I think we have can you see this? This is a slide I didn't show, but this question is getting after like the rotation of the C-terminal domain as it's kind of like rotating up towards open or rotating down away towards in the closed conformation. And a study that we published in 2022 talks about the dynamics of the C-terminal domain quite a lot, and so it does seem like the rotation of the C-terminal domain, like up and more towards open, would be like pulling the N-terminal domain out of the TMD, between TMD1 and TMD2, but we don't see where it goes. And so in the shown in the map, I guess I don't have my shown in the map in both of these structures is the KATP. And this is both the CTD down and then when the CTD is up, but both of these structures are closed. And we can see density for the whole KATP that's like mostly connected between the TMD1 and TMD2. And then when it's withdrawn, it's not clear where it goes actually. So it's not clear if it's down here. Maybe it's like along the membrane, and so it's hard to tell where it's at because of the detergent micelle. But that's a great question, and that's something we want to answer, and we're actively working on. We have an MD simulation collaborator who's thinking about that question, and then we're trying to design mutations where we think that it might be interacting with maybe the N1T2 linker in SUR1 to try to see if we can see where its resting position is. But that wasn't something that I was able to observe in these maps. It just goes away somewhere. So highly mobile, I suppose, or associated along the membrane, and so it's challenging to distinguish it from other micelle density.
Any research ideas?
Can I ask one quick question before you get to the last questions in the chat? I'm sorry if I missed it, but are you using symmetry in these reconstructions? And whether yes or no, like how do you make those decisions?
So
that could be like a 3-hour talk, but
yeah, that's a great question. And I showed two different strategies. I'll just go sit on the second strategy. And the first strategy used C4 symmetry restraints during refinement. And this second strategy was C4 symmetry expansion and focus classification and focus local refinement, focusing on just the core KR6.2 tetramer plus one SUR1 subunit. So kind of two alternative strategies, both based on C4 symmetry.
Thank you. That answers the question.
So following up on the KNTP interaction, we have a attendee wondering if the KNTP interaction with SIR can happen without the help of ATP binding to KIR6.
Yeah, I mean, that's a good question because currently in our I think in all of the structures where we see the KNTP, we have ATP bound, except for this one that I'm showing here, where we have an APO, because I did not include any ATP during this particular purification, and we still see that KNTP bound in the CTD down closed APO structure. So I think that the association is independent of ATP.
Okay, and final question for you: do you see the ATP density in KIR6.2 in the map of the closed conformation?
Oh yes, we do. Mm-hmm. Yeah, and those studies were published in 2021 and 2022.
Oh, great.
And I may have had a picture of it in my later part. Let's see. I guess not. Sorry, but yeah, we've published the ATP density for the closed.
Great, thank you.
I have just a couple of questions. Is panelists not allowed to actually enter the Q&A. That's why we have to ask them separately. This is Mike. Um, uh, two things. One is that these are very good structures with only 15,000 fourfold. Of course, that means 60,000 asymmetric units. That's really very nice structures under 3 angstroms, and they're just really very nice structures. The second thing that I was curious. I've seen very many structures of channels opening and closing and helices moving and rocking back and forth, but I don't think I've ever seen a helix which became a beta sheet. And
Right.
and that's really interesting. It looks like from the movie, it's the helix was part of something that had to stretch out, and it just unwound the helix and made a beta sheet out of it. I mean, I know that there are a lot of structures where actin comes to mind, where certain helices in one actin structure are helices. In another structure, it's a part of a beta sheet. And a third structure, it's a loop. And it's always curious to me when one changes into the other, how do you lose hydrogen bonds? You have to break some and reform some. What's going on there?
Right. Yeah. And it must be like energetics involved, like that it really, once you have one secondary structure, it requires a lot to kind of move it into the other one to transition from like the open and closed states. And we kind of see some of that when we look at the electrophysiology of the bursting currents. It'll like be open, and then kind of more close, close, close. And so it's like maybe that could be one of the explanations for that, is that you do have to break and reform many hydrogen bonds.
It's an energy pump you have to go over, and you don't do that. You don't do that.
Yeah.
With impunity. You gotta do something to the structure, and and that's great. It's stable in one and stable in the other, but the transition is tough to do.
Yeah, that's absolutely true. And there are these like very large domain motions that are also occurring with that, maybe helping to drive that. But yeah, it was a surprise to us to see these kind of structural rearrangement, and to see like the SGR1 actually becoming part of the continuous beta sheet of the C-terminal domain, since it seems like it would be so stable, it would just kind of lock it in that open conformation.
And it could. I mean, it's tough to do to the transition, and maybe it should be tough. And that's and one more thing about different conformations and locking, looking at one and ignoring the rest, and looking at the other and ignoring the rest. The Gaussian mixture model that's been developed by Miljan, who works at Slack now, and that is really good at isolating those different ones, concentrating on one area of the structure and letting the other part do what it wants to do, but you get a good high resolution structure of each of the pieces, and then it's your job to knit them together at the end. But it gives you a good way of doing refinement on individual parts of it.
What was that called again, Mike?
The Gaussian mixture model, and there's several papers on it now, and it's a good it's a smart way of doing things without hard masks that isolate one, or it. Yeah, it's an intriguing way of doing things.
Yeah, that's a great suggestion. I'll look at that. Thanks.
So I think you have fielded all the questions you are required to.
Yeah, thank you for sticking on an extra couple minutes to let the panelists carry on with our questions.
Yeah.
Yeah, great. Thanks. Yeah, thanks again. It was a nice discussion.